So people see where we're going. So we've now talked about gathering data on individual fitness. Now we take that data with measurements of a trait or traits of interest. We'll talk about multiple traits, that is multivariate traits, tomorrow. But for now, we're just going to start out by talking about a single trait. So here are some of the basics. We'll talk about some of the basic concepts. We'll talk about the notion of a fitness surface versus a fitness landscape. This describes how individual fitness changes as I change a phenotype. That describes how population mean fitness changes as I change distribution parameters. We'll talk about measuring changes in distribution. So I have a trait, I measure it, I have an episode of selection, I reweight the trait, the individuals, and then look at the fitness-weighted distribution of trait values, and that gives me indications of selection. We'll talk about measures. We've talked about S, the intensity selection. We'll talk about beta. Beta was a gradient. This is different from the Bateman gradient. These are for changes in means. We'll talk about C and gamma for changes in variance. These, for a single trait, are basically scale versions of one another. When we talk about multiple traits, the distinction is quite important. S and C are direct measurements that you see. Beta and gamma in multiple traits are ones that adjust for correlations among your measured traits. So we'll talk quite a lot about that. We'll also talk about a particular metric which looks at the change in the entire distribution. Then we'll talk about some examples. Okay? That's kind of where we're headed. So blah, 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 blah. Basically, what I said before. You, right? Blah, blah. You basically, when I want to ask if selection has occurred, I have individuals that are measured, and then I have an episode of selection. It could be like Bumpus's classic case where individuals, basically had dead, so you could have dead individuals and live individuals, that's your sample. And what I basically do is I take the entire group before the episode occurs, so I measure everyone. I get a distribution, typically a mean, a variance, maybe the full distribution. Then I weight those individual values by their fitnesses. In the simple case, weights are 1 and 0. Some survive, some don't. And I can compute a mean on that survival set. In more general cases where I have fitness weights, I then adjust those individuals, and I can compute means before and after an episode of selection. And that's what we look at. And this enterprise basically goes back to the first couple of studies. Number 1, Weldon, I love this. So look at that centrally look. And the caption says, yours affectionately. It's like, yeah, right. Come on now, right? Yours affectionately. He single-handedly held back the synthesis of Mendelian genetics and Darwinian evolution because he was such an a-hole, to put it bluntly. No one liked him. It was only when he died that the people on different sides realized we can actually get along. But that's neither here nor there. One of his early works was looking at the carapace length on these green crabs. And what he noticed was that these green crabs seem to have their carapace lengths go down in a harbor where they just put in a breakwater. So it was one of the first studies where you actually measured a trait and saw it changing over time. This was revolutionary. This was 1880s, 1890s, just after Darwin had been, a couple of years into Darwin. And he also got Carl Pearson interested in statistical methods. So a lot of modern statistics traces back to early attempts to look at genetics. So regressions come from heritabilities. Correlation coefficients trace back to early attempts to analyze selection. He also did something very interesting. He basically showed the carapace length was related to silt size with a direct manipulative experiment. So Weldon was one study. The other study was Bumpus, I talked about. Here's the house sparrow. So Bumpus was walking along and saw all these frozen sparrows after an ice storm, measured a whole bunch of stuff, took them back home, measured a whole bunch of stuff. Half of them lived, half of them died. And this data set is still analyzed today. So it shows you the advantage of publishing and making public a very well-gathered, careful data set. So both these individuals were able to show that survival seem to be correlated with differences in certain morphological characters. So the first attempts to actually get a handle on, do certain traits potentially influence fitness? What I just said, right? So basically, here's Weldon. So he had 136 and 72 survived. So that's actually not too bad. So how would you detect selection in these type of samples? So you have the sparrows, you measure everybody, then you measure those that survive. Well, number one is you compare the mean of all the sparrows and then the mean of the set that survived. That's an easy weighting because you basically look at the subset over here that survived. If we're different number of mates, we'd go ahead then and weigh those individuals by those PI values I told you before. You weight them by their initial frequencies times the relative fitness. So I can get a fitness distribution of trait values, a distribution of trait values beforehand, and then compare that to a fitness-weighted distribution. And for example, look at change in the mean or change in the variance. Most of the focus is on change in the means because people are interested in, will that character mean change over time? Relatively more recently, people have been concerned too about change in the variance. If I have stabilizing selection, so individuals with some optimal value or most fit, if the population is sitting at that optimal value, the mean won't change much, but the variance will shrunk down as outlier individuals are removed. So quantifying changes in mean and changes in variances are important, but as I mentioned, other factors besides selection on the focal trait can result in a change in the moments. So what are some of the complicating factors? Ontology, growth. So for example, I measure a whole bunch of trees. I come back next year and measure the trees that have survived. Those trees have grown. So you have to be careful that the differences you see aren't simply due to the traits changing over time. But the biggest problem is selection on a correlated variable. So the biggest problem is selection doesn't occur on your trait, it occurs on a variable that's correlated with your trait. So for example, let's imagine I think selection is on body mass, but in reality selection is on height. Biomass, so body mass and height roughly correlates. So what you basically see is taller individuals would have, heavier individuals would have higher fitness. They have higher fitness not because they're heavy, but because they're tall, and tall people tend to be heavy. There's a variable in there that unless you include it, you're going to get a false association. We'll talk tomorrow about how you include variables and how you test for whether or not you captured a lot of them. So here's an example. Suppose you're an evolutionary ecologist and you're looking at a plant and you measure some chemistry and then you measure seed output, and what you notice is plants with higher levels of alkaloids produce more seed. Immediately, you cleverly envision that this is because alkaloids give you protection against insects, and therefore the plants with higher levels of alkaloids have a lower insect load on them, therefore they make more seed. It sounds wonderful, but suppose here's the situation. Here is alkaloid level, here's seed production. These are plants in low nitrogen. Low nitrogen gives you soil, gives you low alkaloid levels, low seed production. These are plants in higher nitrogen soil, gives you higher alkaloid levels and higher seed production. Within each group, within the triangles, the regression is flat. Within the circles, the regression is flat. When you put these traits all together, you see an association. This is a very simple case with a single hidden environmental variable, but it kind of makes the point that you could have a trait that's involved, or you could have an environmental feature that's involved. An environmental feature means it's something that a parent can't pass on to its offspring. So that could give you a marker trait, I keep on saying marker. That could give you a fitness trait association that's not driven because differences in that trait value cause differences in fitness. If you could measure that trait at the same level of nitrogen, you'd see no impact, but rather you have a confounding when you have high nitrogen, you tend to have high alkaloid levels and high seed production. So missing traits are a real problem. Let me take an aside here really quickly and talk about multiple trait selection, that is, two-trait selection. This expression here, because they're in bold, is the multivariate Breeder's equation. What's the Breeder's equation? R equals h squared times S. What is h squared? Additive variation divided by phenotypic variation. When I have a vector, a list of traits, what I want to do is relate the within generation list of changes, that vector S, the selection differential, with the across generation changes of the trait mean R. So R is a vector or a list. The fifth element is how much trait 5 changes after one generation, given the strength of selection, the within generation change on all the traits. G and P are matrices, a rectangular array. In G, on the diagonal are the additive genetic variances. On the off diagonal are the additive genetic covariances. And what that means, a positive covariance means if you have a good breeding value for height, you tend to have a good breeding value for weight. Remember, breeding values are important because if I take the breeding values of parents, the average of those tells you how much above or below the overall mean the offspring are going to be. So this is what's called a genetic variance covariance matrix. P here is a similar matrix, but on the diagonal are the phenotypic variances, and on the off diagonal are the phenotypic covariances. So do traits phenotypically covary? That could happen because they share genes, or within the same individual they share an environment. So if you get more nutrition, you're both bigger and taller, not because of genes, but because you shared that environment. So basically, this expression here tells you how characters change. This is often written simply as G beta. Beta is called the selection gradient, so I can write R equals G beta. That's called the Landy equation. And G beta is P inverse S. Remember, one way to think about an inverse variance covariance matrix is it undoes stuff. It removes correlations, and it basically gives each trait the same variance. So what beta here does, we'll talk about this a lot more tomorrow, beta tells you which of your traits are actually under selection. If trait 4 has a beta value that's positive, there's selection to increase that trait. If trait 4 has a beta value that's negative, selection to decrease that trait. Trait 4 has a beta value of 0, no selection. S, the change in trait 4, could be quite different from the change in beta. And for two characters, here is what we can write S as. So for two characters, we can write the within generation change in character 1 is direct selection on character 1. So P11 simply means the phenotypic variance of trait 1, plus P12 means are trait 1 and trait 2, are their phenotypes correlated? And if so, then S will also reflect the selection on trait 2. This is your key equation. Suppose S is 0. And you have two traits, trait 1 and trait 2. Well, if trait 1 and trait 2 are not correlated, then the sign of S tracks any direct selection on trait 1. However, if trait 1 and 2 are trait correlated, and both traits are under selection, the sign of this may be different from the sign of that. This could be positive, that could be negative, but this quantity here is sufficiently positive to make S positive. This could be 0 because that's positive and that's sufficiently negative, or vice versa. This could be negative when that's positive, but that's negative. So the point is, the within generation change in a trait depends upon selection acting on that trait and selection on any phenotypically correlated trait. So phenotypic correlations are something we need to worry about. And again, we'll say a lot more about this tomorrow. But the point I simply want to mention here is just because you have a within generation change in one trait, that could arise from direct selection on that trait, or it could arise from direct selection on one or more correlated traits. such that the sign of S1 really tells you nothing about the nature of direct selection on that trait. It's direct selection plus selection on any correlated traits. That's kind of the key idea. I can also ask, how does trait 1 change across generations? And trait 1 changes across generations. An expression looks very much like this, except I replace S by R, and I replace phenotypes by breeding values. So the response in trait 1 depends upon the direct selection in trait 1 times the additive variance in trait 1, and the direct selection in trait 2 times the covariance between trait 1 and trait 2. So one thing you might envision is one way to get around this problem of whether or not trait 1 is under selection is to ask, does trait 1 change across generations? Because across generations don't involve phenotypic correlations. These phenotypic correlations are how traits covary within a given individual. But across generations, trait 1 can change because it's under selection, and or it can change because a trait that's genetically correlated. Genetically correlated means the breeding values for one trait in you somewhat predict the breeding values for another trait. If they're positively correlated, if you tend to increase in trait 1 in your offspring, you tend to increase in trait 2. If they're negatively correlated, when trait 1 goes up in your offspring, trait 2 goes down. So a change in the next generation could be because of direct selection, or it could be because of correlated responses, and it's both of these. So just because R1 changes in the offspring tells you nothing about whether trait 1 is under selection, nor anything about the sign of that. So just because a trait shows a response to selection doesn't mean it itself is under selection. Within a generation, a within-generation change can be driven by phenotypic correlations on other traits, and a cross-generation change can be driven by genetic correlation on other traits. So that's the concern you need to worry about when you have multiple traits. So here's an example. I think I mentioned this. So Lisa Cook had an example where she looked at antler size in red deer. Males with large antlers enjoy increased lifetime breeding success. That beta there is highly significant. Let's look at what that beta means. Basically, that's the change in mean. That's highly significant. Antler size is also highly heritable. So despite selection and heritability, antler size, it says no observed response. Response is actually negative. It's not sufficiently negative. The authors suggest that the reason we see this factor here of this association between antler size and breeding success is that males with large antlers and good fighting ability, which gives you more mates, are a function of increased nutrition. Increased nutrition gives you bigger antlers and larger fighting size. So all this component here is based upon a non-heritable factor of antler size. That is the environmental component, not the genetic component. That's the concern. Unmeasured characters or unmeasured factors. Because an environmental feature really isn't a character. We'll call it an unmeasured factor. So given that concern, how can we start to describe fitnesses? So we can talk about the expected fitness. WZ is simply I take all individuals with a certain phenotypic value and I look at their expected value. That generates a curve, right? If I do a plot of WZ versus Z, it could be a line, it could be a hump, could be some complicated structure. We call that a fitness surface or a fitness function or a fitness profile. We can also talk about the relative fitness surface, which is simply that same curve standardized divided by W bar. A population, so the importance of this, yeah. So here are potential examples then of such individual fitness functions. Here's Z. I put the mean on here for reasons we'll come to in a second. And here's the fitness. So if I see something looks like this, If the population mean is around this hump, we say the population experiences stabilizing selection. As you move away from the mean, the fitness declines. If the mean is sitting right here, and instead of having a maximum, I have a minimum, I have disruptive selection, that intermediate values are not favored, whereas extreme values are favored. A situation here where the fitness increases as I move along in Z. You notice it's not a line, it's a curve, but it's increasing. We call that directional selection, or I could have some combination. And people often talk about stabilizing, directional, and disruptive, but that's a bit misleading because whether or not what you have in some sense depends upon how wide the population distribution is. For example, if the population distribution is really narrow, then at any point here, I'm only experiencing kind of one type of selection. If the population distribution is broad, I can experience multiple ones. The key concept here is simply that we have these surfaces, and then people talk about individual fitnesses having peaks or valleys. Peaks correspond to potential regions of stabilizing selection. Optimal phenotypes, as you depart, fitness goes down. They can have valleys. So optimal, you have a minimal phenotype, the least optimal. As you move away from that, you increase. And of course, they can be much more complex. The key part is the original way people describe fitness surfaces was with a quadratic regression. Quadratic regression captures this, but not that. Quadratic regression, no matter how many variables, you only have one maximum or one minimum. And as we'll see, that's a constraint we have to kind of work around. So the way people describe those is stabilizing selection and disruptive selection. If I have a negative curvature, we more generally call that as having a concave fitness surface. If I have upward curving curvature, we call that a convex fitness surface. So this notion of stabilizing disruptive is often now replaced with some idea if I've got a negative curve on that fitness surface or a positive curve. And obviously that breaks down when you look at something like this, but this is just for looking at curves which have a bit of curvature which only have one extremum. So for example, over here, that's not a linear fitness surface, that's slightly curving downward, and so that surface there would be the same as stabilizing. We call that stabilizing section because we'd have a concave curvature. That's individual fitness surface. One way to move away from individual fitness surface is to then talk about what happens over an entire population. So fitness surfaces are used when we map individual fitness as a function of phenotype. A fitness landscape is basically described by, here's our individual fitness surface, and now I look at mean fitness as I change the mean or change the variance of that. And we call that, so here's an example. Here's an individual fitness surface, very wobbly, very messy. Here's a distribution. I then ask what happens with this distribution to the mean fitness as I change it. So I simply take this curve and move it over and integrate under that curve. This gives me then a mean fitness surface. And we use the notation typically. We call something involving individual fitness a fitness surface. We call something involving mean fitness a fitness landscape. A fitness surface is what an ecologist is interested in. It tells you how trait values change fitness. A fitness landscape is something an evolutionist is interested in. It basically tells you how the population is going to evolve over time. So we have this notion, individual fitness surface involves individual fitness as a function of Z. The mean fitness surface, which preferably called a fitness landscape, basically tells you as I change the population mean, or maybe some other parameter like population variance, how does mean fitness change? And it's these fitness landscapes, concept dating back to Simpson, who called it an adaptive landscape. It's these that basically allow us to ask how the population is going to change over time. I won't say much about that connection. Chapter 24 covers all that. But basically, the key part is this distinction between a fitness surface, individual characters, how they change as a function of phenotype and a fitness landscape for the entire population. How does the mean fitness for that character change as I change the distribution? So that latter one, the fitness landscape, requires not only individual fitness surface, but requires the distribution of values. So for example, if this had a broader variance, I'd have a different curve here. People, when they talk about a fitness landscape, generally assume you're only changing the mean as you move along, but in theory you could change any part of the distribution you want. So fitness surfaces for individual traits, fitness landscape for mean population fitness. How does individual fitness change as you change character? How does mean population fitness change as you change mean? So yeah, not really. What the concept of getting that is something called the genotype by environment interaction, which is, so the other way to get around that would be on that same curve, plot multiple curves. Here's the fitness function in environment 1. Here's the fitness function in environment 2. I think that makes a lot more sense visually, a bit easier to interpret. So the questions about, yeah. Okay, large variance. So let's think about that a little bit. So we're not going to talk much about this. We can talk about the idea. So highly plastic means you've got a large environmental variance. Potentially. So if you have a population under stabilizing selection, you want to do two things. Move the mean under the peak, shrink the variance. Because if you think about it, if you're under the peak, if you have a broad variance, you've got more individuals on the lower fitness tails. So what stabilizing selection would do would be to move the population mean towards the middle and shrink the variance. In contrast, disruptive selection, you've got a couple scenarios depending upon the dynamics. Number one is you move to one side or the other of the peaks. Number two is you go in the middle, but you've got a very large variance. Large variance means you then have individuals sitting over here, which are higher fitness. So if you basically have phenotypic variance you can select for, which is often the case because it's heritable, you're going to shrink it when you have stabilizing selection. You're going to blow it out when you have disruptive selection. Now, there's a third issue, and the third issue has to do with kind of bet hedging. And if you have outlier environments, and in that case, the conditions under which you select for higher environmental variances depend upon the particulars of the system. Chapter 17 talks about that. Yeah. So the variance, remember, is caused by two things. Number one, environmental variance. Number two, genetic variance. Under stabilizing selection, you generate negative disequilibrium that shrinks your variance, but that's easily removed. If your peak shifts, you can track that. So you don't. It's a long time scale to lose variation under stabilizing selection. See chapter 28. We'll talk about that in a lot of detail. Yeah, so if you're interested in, so chapters 16 talks about changes of the additive variance under stabilizing selection. Chapter 17 talks about heritable changes in the environmental variation. And then chapter 28 talks about how variation is lost or maintained under stabilizing selection. 28 is long and very complicated, but the others are a little bit shorter. Your roadmap moving forward. It's like citing biblical verses, like Corinthians 4, it's like Walsh 17. Yes. Absolutely. And we'll show examples of that, basically, if your fitness surface is a saddle, you can have that. And one of the things we'll talk about at length tomorrow is how do you visualize, even in quadratic space, these complicated fitness surfaces? It's not easy. It turns out the answer is looking at the eigenvalues of the gamma matrix, which we'll talk about. Yes? Uh-huh. Yeah, so great question. So basically the distinction is right now we're not using any aspect about the genetics. If I want to project how the population is going to change over time, I'll need to know something about the genetics and then use derivatives on the fitness landscape. For now, we're simply making this distinction. If I want to ask how character fitness changes as a function of trait value, average fitness, that's a fitness surface or an individual fitness surface. If I want to ask how the population mean fitness changes as I change the population mean, or maybe something like the variance, that's in a fitness landscape. So individual fitness surface, population landscape. Yeah, no, the adaptive part tells you what direction selection is forcing you to move. Whether you move there or not depends upon if you have sufficient genetic variation. Huh? Good question. I appreciate these questions for clarification because if you're a little uncertain on something as we start, I find I tend to, when I have that similar problem, I tend to keep that uncertainty bouncing around and I try to resolve it and I lose track of the lecture. So if there's things you need clarification on, always asking early in any class is always highly recommended. It's a good way to follow the lecture a bit more carefully. So that's kind of an overview of some of the problems, a little bit of the history, and this notion of fitness surfaces and landscapes. Now let's look at some of the specific measures we have to measure changes in phenotypic, the distribution. The first one is phenotypic means. Here are two quite contrasting figures in evolution. There's Alan Robertson, long distinguished history at Edinburgh. Like I said, he's kind of like the Tesla of quantitative genetics. George Price, one of the most fascinating and enigmatic figures in evolutionary biology. Anybody know exactly how many papers he wrote during his career? 4. 4 of the most fundamental papers. They're all short. So how many of you have used evolutionary stable strategies? Price and Maynard Smith. Fisher's fundamental theorem is an esoteric thing about how mean population fitness changes. Nobody knew what it meant. Price figured out what it meant. Then he had the Price equation in 2 papers. So Price was a chemist who worked on the Manhattan Project. Then he went to work with Cedric Smith at University College London from I think 72 to 74, like a 2 or 3 year period, had this incredible work, dropped out, gave all his belongings to homeless people, and died of squalor 2 years later in the poorest area of London. So obviously he had mental issues. There's several interesting books. One book is called The Prince of Altruism, because what Price resolved was, how do altruistic traits evolve? I'm a rabbit. Actually, make it more interesting. I'm a pika, a rabbit relative. I'm sitting on a lovely glacier, looking around with my fellow pikas, and all of a sudden a giant hawk squeaks by. Individual fitness says, get your ass under a rock right away and don't call attention to yourself. Alarm calls say, look out guys, there's a hawk, calling attention to yourself. How do those traits evolve? Well, Price showed the conditions under which they can evolve even under individual fitness, and he had this general framework called the Price equation. Of course, he didn't call it that. But before the Price equation came out, in 1966, and then here are the 2 Price papers, and so they're 70 and 74, the 2 Price papers, they had this identity called, I now call the Robertson Price. By the way, this is an interesting paper because Robertson is usually extremely clear. This 1966 paper had the enduring title of A Mathematical Model of the Culling Process in Dairy Cattle. It is incomprehensible I know literature pretty well. I can't figure out what the hell he's saying. I'm not sure what happened then, but usually his writings are pretty clear. Anyhow, they basically showed that the within-generation change completely generally can simply be written as the covariation between relative fitness and trait value. The reason this is nice is you can take very elaborate systems where it's kind of not clear how you weight stuff to get a within-generation change. If you know relative fitnesses and phenotypes, that gives it to you right away. This is a special case of the Price equation. Price did this much more generally, asking not how the within-generation change happens, but rather how systems evolve over time. The Price equation, see Chapter 6, is a completely general expression for how anything evolves over time. It's completely general, and it's largely very hard to apply because it requires you to know things that basically is based upon covariances. But it's a framework for how a lot of people think about complicated questions in terms of how behavior should change under cultural evolution, how behavioral traits can change. So this is kind of the tip of the iceberg. And the way you get this simple expression is pretty straightforward. If I define the frequency of Z after selection, it's pretty simple. Here's the fitness weight. So there's the distribution beforehand. Here's the fitness weighted distribution. To make sure that normalizes to 1 and it's a proper probability distribution, I divide by that. That's W bar. So what this basically shows is the fitness weighted distribution is simply the relative fitness times the distribution beforehand. What is the mean after that? Well, the mean is simply the average value over the distribution. So here's that distribution. You can write it this way. If you look at this expression here, Z times WZ, that's the expectation of this product. That's the first part of the covariance. So there's that first part. So what is S, the within-generation change? It's the change after selection minus the change before. We know this is this product here. U is expected value of Z. Expected value of relative fitness is 1, so I put in there, and that's the definition of a covariance. So very simple definition, very simple proof of the Robertson-Price identity, and it's extremely powerful, and there it basically is. The within-generation change is simply the covariance between relative fitness and phenotype. And that's kind of a foundational piece we'll use for the analysis of selection. It's kind of a foundational piece for looking at general selection theory, Robertson-Price identity. So directional selection, whether there's a linear trend in that data, we can have several different ways to measure that. I think I mentioned that. So one way to measure that is the directional selection differential S. We've called that the selection differential. We're going to call it now the directional selection differential because we'll have a corresponding differential for changes in variances. So S is the observed within-generation change in the mean. The selection intensity is simply that standardized, so that change in phenotypic standard deviations. And finally, the directional selection gradient is simply S scaled in terms of variances. Now you look at those three, you go, what's the big deal? They're all the same. One's by itself, one's divided by the square root of the variance, one by the variance. The difference is that's in univariate traits. When you go to multivariate traits, this actually involves a matrix product and is quite different. And as I mentioned before, the directional selection gradient is the observed within-generation change in a particular character. The directional selection gradient is the actual selection acting on that target, which can then, because of phenotypic correlations, give you very different value of S. So those are three metrics that we'll use. We'll mainly talk about two: directional selection differential and directional selection gradient. One, again, for a single trait, with only one trait acting, there's scaled versions. For multiple traits, one's the change you observe, the other S, the other beta is the actual amount of direct selection acting on that trait when you remove the phenotypic correlations that adjust for that. So those are all measures of change in the mean, right? S is the measure of change in the mean. We can also have measurements of change in the entire distribution. why just look at the mean? And how many of you have heard of the Kolmogorov-Smirnov test? Sounds like a great vodka, right? And basically, the idea behind the KS test is this. I have some empirical distribution, cumulative, and then it basically goes up to 1. So basically, this here, let's say this is 10, that's the probability of having a value 10 or less. That's one distribution. Then you have a second distribution. And the KA test basically looks at that difference between those two curves and standardizes to give you a test statistic. It's a non-parametric test. It's not very powerful, but it's nice to use. So it's a general way to compare two distributions. I'm not looking at the mean, the variance, I'm simply comparing their cumulative shape. You can get a corresponding thing, which is called the distributional selection differential. So not directional, but distributional. It's called DSD. It's a fairly new concept. Basically, here's the idea. This thing here, are these things? That's just the cumulative probability. So f of z is the probability. So for example, f of 10 is the probability of having a value of 10 or less. F star of 10 is having the value of 10 or less for the selected population. So this absolute difference here is just this shaded part I've shown difference between the two curves. That then is a metric for how different the two distributions are. And that's one way of estimating. Again, you want the estimator, look it up in the notes. And the key part about that is if WZ is a monotonic function, monotonic simply means that it's always increasing, like a line. It doesn't have a flat spot, it doesn't dip. If you have a monotonic function, then Hinshaw and colleagues showed that D equal the absolute value of selection intensity. So it's a nice connection. But they showed something else then. It says you could take the value that you observe D for your thing, subtract off the selection intensity, and that tells you what fraction, the so-called non-directional selection fraction, what fraction basically is due to curvature, basically, departures from linearity. You can also use D, and D can be bounded by the variance and fitness. So I simply mention this because if you don't want to focus on a specific mean or variance that is a specific moment, this is a metric that's been introduced that allows you to look for shifts in the entire structure of the distribution. So DSD, how widespread it will be is unclear, but it's an interesting metric, so I thought I would include it. Yeah, question? Yes? Great point. So the black curve is my distribution of the entire population. So cumulative distribution, right? So like a histogram, I just sum the histogram, make it bigger and bigger. The red curve is now I take the fitness weighted values. So Z is the distribution before selection, Z star is the fitness weighted distribution. So the simple case, alive or dead, the weighting is, it's just a distribution of the subsample. If I've got differential weightings among the survivors, then I would put those in as we talked about in the first lecture. So we can talk about measuring changes in the mean with a couple of different metrics. We can talk about this, which basically talks about how the whole distributions change. I want to say a couple of things now about directional selection differentials and directional selection gradients. So S and beta. The change in mean within a generation can be measured by a couple of things, and also you can do the intensity, but this is an important connection I want to talk about. So beta equals S divided by sigma squared Z. Why is that important? Let me use the board over here. And I'm trying to go back and forth so people don't, miss out on that. If I have a regression, X, Y, some intercept A, some slope, for one unit change here I get a certain slope B. So my regression is Y equals A plus bx. What's the expression for the slope? Beta equals the covariance of y and z divided by the variance of x. That's a general expression for the slope of a univariate regression. The covariance of the two variables divided by the variance of the predictor variable. What is this? That's the covariance between z and w divided by the variance in z. That's the slope of the regression of relative fitness on phenotype. So that's a nice interpretation, and that interpretation holds when you move to multiple characters as well. We just define this ratio a little bit differently. So the directional selection gradient has this nice interpretation because it basically says a beta value says so a beta, for example, of 0.2 says a 1 unit change in x changes relative fitness by 0.2. So it's a metric that basically gives you some idea for how changes in x translate into changes in fitness. So beta is the slope of the regression of relative fitness on the trait value z. I can write the regression this way. I can then use the trick that the regression line always passes through the mean value of the two variables. If I use that trick, I can remove the intercept, and I can basically say w equals 1 plus b times the quantity z minus the b. So beta here basically tells you how sensitive relative fitnesses change when I change my trait value. It's got a nice interpretation. A negative beta means the trait goes down, and the size of beta tells you how important it is. So for example, a beta value of 0.1 means a 1 unit change results in a 10% change in relative fitness. People usually standardize z first, so z has a variance of 1. In that case, a beta value of 0.2 says a 1 standard deviation change in the trait changes relative fitness by 0.2. So when people report beta values, they've often standardized the trait to have a unit variance, and that allows you to compare betas. So if I've got 4 traits, each beta value says a standard deviation change in that trait changes relative fitness by the amount beta. So it's a nice interpretation. So we're talking about a univariate case. In a multivariate case, this is P inverse S, which takes care of that, takes that into account. Now, there's still some normality assumptions I'm going to gloss over. Who's going to come back and get around them when we talk about how you handle non-normal residuals in fitness. We haven't got there yet. Yeah. We'll come to that. So one of the issues, if you're working with Hull and Hansen, for some reason you might think about variance versus mean stabilized. We'll talk about those at length. So the different ways to standardize this, and there are different consequences we'll talk about a little bit later on. So this is the Landy equation. R equals beta times the additive variation. This is basically rewriting the Breeder's equation. And what this basically gives you for a single trait is it says a trait will respond, A, if there's selection on it, and B, the amount of response will depend upon how much additive variation we have. But the really interesting thing is that beta has another interpretation. We keep on calling it a gradient. Remember from calculus what a gradient is? If I've got a multidimensional function and I'm sitting right here, the gradient vector is a vector of partial derivatives. And basically it's a vector that points in the direction of steepest change. Steepest increase or steepest decrease, depending upon what the function does. What you can show, and show in the notes is pretty straightforward to do, is that if the traits are normally distributed, and again I'll have a table tomorrow that puts all this stuff together. If the traits are normally distributed, then beta is equal to the log of mean fitness divided by mu. That's a fitness landscape, right? How fitness changes as I change the population mean. So this response term here, the character, how it responds depends upon the fitness landscape, how relative fitness changes with mu. But beta is also a measure here of how the individual fitness surface changes. So we'll talk about these connections a bit later on. We can talk, though, about beta saying how individual fitness changes as we change trait. Beta also then tells us how the overall trait means change, and that depends upon this fitness landscape. Yeah, question? Yeah, so what you do is, if you think about it, what's the derivative of a log? 1 over variance what's inside. Same thing, right? That's why you get those expressions. So you can also write it as log relative fitness as well. Yeah, good point. And again, all these connections I'm going to hint at here, which may be a little bit hard to follow, we'll come back and really cement them when we talk about multiple traits. Because, you know, beta and S basically seem to be scaling of each other with a single trait. With multiple traits, it means something quite different. And that all traces back to this notion we talked about here. If I've got 2 traits, for example, what I observe for the within generation change depends upon the beta. So now you see that beta 1 and S1 are actually rather different. So when you have multiple traits, what you want to look at are the betas. S is what you observe. The betas tell you what the actual targets of selection are, assuming that you've got all the traits for selection in your data. And again, that's something we'll chat about. So here's our summary of within generation change based upon the mean. I'll mention we talked about this distributional selection differential, which is a general metric for asking how different 2 distributions are. I won't say more than that. Then we have 3 different measures. The directional selection differential, S, the observed change. The selection intensity, that's kind of a standardized version, just in standard deviations. And then the selection gradient. Again, these 2 simply look like scalings, and they are for a single trait. For multiple traits, they change. This has a nice interpretation. If Z is normally distributed, the trait's normally distributed. Beta is the gradient with respect to the fitness landscape, how the mean fitness surface changes as I change trait value. Likewise, beta is the slope on the best linear slope on the regression of individual fitness on trait value. So I can interpret under normality, and again we'll talk about this, beta as either a gradient with respect to a landscape, and that tells me how traits evolve, or I can think about beta as telling me what a single unit change in a trait value tells me about how the relative fitness of that trait's going to change. So these are the common metrics. We talk about directional, we talk about change in mean, we talk about a selection differential for an observed change, we talk about a selection gradient for something that will be able to, in multiple traits, tease apart direct and indirect traits. So we have a directional selection differential S, a directional selection gradient beta. Okay? The reason I mention that is we're now going to talk about changes in variances, quadratic effects. But before I do, questions? Yeah? That's the before selection, that's correct. So the variance in the trait before selection. Let's now look at changes in the variance. Selection can cause the variance to inflate, deflate, or not change at all. So initially you might think, oh, you know, the change in mean, when you compute that, you should take the fitness-weighted mean after selection minus fitness-weighted mean before selection. So obviously for change in variance, it's simply the variance after selection minus the variance before. The problem is Landy and Arnold, so this is the key paper, Landy-Arnold 1983. This is the paper that basically talked about, so the Arnold and Wade papers in '85 talked about partitioning fitness and opportunity for selection. The Landy and Arnold paper talked about these actual estimates based upon trait values. So Landy and Arnold is basically associating traits with fitness. Wade and Arnold and Wade are basically partitioning fitness up to some extent independent of traits. So what they showed was, what is this difference of variance after minus variance before? What is that? Well, you can show, and again, the books show this, that's equal to the covariance of relative fitness with respect to square deviations from the mean minus the selection differential squared. Well, this is kind of what you want, right? This basically asks, as traits get further and further from the mean, what happens to fitness? If this term is positive, it means that as you move further and further away from the mean, the fitness goes up, disruptive selection. If this term, covariance is negative, it means as this gets bigger and bigger and bigger, that gets smaller, that's stabilizing selection. If there's no association whatsoever, no quadratic association, the change in fitness is minus S squared. So directional selection by itself reduces the variance, so you have to adjust for that to compute the within generation change. Recall, what was S? S is the covariance of W and Z. If I scale it to have the mean of 0, which is easy to do, this would be the covariance of W and Z squared. That seems to be the natural metric for the quadratic selection differential, and in fact, that's exactly how it was defined. So this is the quadratic selection differential. It's the change you see corrected for S squared. Why? This is really what we want. That's a Robertson price-like term for quadratic effects. Remember, for change in mean, it's W and Z, the covariance of those. And so what you basically do is you take that observed change and then you correct for the fact that any directional selection is going to decrease the variance. So this is your quadratic selection differential. And you get this funny thing because if you have directional selection, it deflates the variance. So Rosemary and Peter Grant, here they are with their, I think it's Royal Society of London medals. They did a huge amount of work on Galapagos finches. There's a Galapagos finch. And they studied populations on these islands for years and years and years. Some of the most interesting stuff in evolutionary biology are these things. So what you can basically do is, what they observed was they had a severe drought on the island of Daphne Major, and in, I forget which year, they had a couple of droughts they followed. In this particular one, the mean and the variance in body weight for 640 adults were these values before the drought, and after the drought, only 85 survived. So a big drop-off. Here's what the means and the variances are. So you notice here, the means change a bit. The directional selection differential is 1.06. The variances are basically almost the same. However, if you use this value, you haven't accounted for the fact that you haven't adjusted for S. So when you compute the quadratic selection differential, that's the within generation change, that's S squared. It's quite substantial. And not only substantial, but the standard error shows it's significant. So if you didn't make that adjustment, that change there, from here to there, would imply there's almost no quadratic selection. But in fact, there was actually almost as much, in fact, equally strong, like slightly stronger selection to change the variance as compared to change the mean. What it generated was disruptive selection. A positive value that means you inflate the variance. That means as you move further away from the mean, your fitness increases. And the idea is, in a drought, food resources are limited. If you partition your population into large seed versus small seed, there's more food to go around. That's a rough way of thinking about that. So if you didn't make this correction and simply looked at the within generation change in the mean, you'd assume that almost all the selection, that is the structure of that individual fitness surface was largely linear, so the mean shifted. What you see here is, in fact, there's also a significant curvature in that, and the curvature in that is upward, so individuals who move away from some value, as you move away, fitness increases. If it was a negative curvature, as you move away, fitness would decrease. Landy and Arnold called C the stabilizing selection differential, but for a variety of reasons, quadratic is better, and one reason why is just because you've got negative curvature does not mean you have stabilizing selection. If your curve looks like this, so it's not linear, it's curving down, you've got negative curvature. But unless your population experiences the hump, your population is around that peak, it's not formally stabilizing selection. Unless your population is around a valley, that is, the range of your population includes that valley, it's not formally disruptive selection because your population doesn't see that. So because of that, people now call this the quadratic selection differential. So a reduction in variance by itself is not an indication that convex selection is occurring, and an increase in variance, so people use the term concave and convex to basically describe that. So convex selection means I've got a negative curvature, stabilizing selection means I've got a negative curvature, and the maximum is within the range of phenotypes seen in the population. Concave selection means I've got a positive curvature. Disruptive selection is positive curvature, and the minimum is in the range of phenotypes seen. So people often talked about this as a stabilizing selection differential. It makes more sense to talk about it quadratic and ask if it's a positive term, it's concave, a negative term, it's convex. With these special cases occurring only if you have negative curvature and the maximum occurs in the range seen, or positive curvature and the minimum occurs in the range seen. So that's how we describe the sort of curvature of these surfaces. We can do some more things with C. So writing C here, remember C now is this Robertson price-like term. So what I can then do is show that C is bounded by this expression here. That's the opportunity for selection. This fourth moment term is this expected fourth moment here. Mu, x minus mu, take the fourth power of that, take the expected value. You might recall that's called the kurtosis. The kurtosis basically tells you, do you fall off, your tails fall off quicker or slower than a normal? For a normal, the kurtosis is given by 3 times the variance squared, or 3 sigma to the fourth. So if you assume a normality and plug that in, this thing collapses to here. So what that basically tells you is your absolute change in variance, standardized by that change in variance, is bounded above by twice the intensity of selection, the opportunity for selection. So just like you can bound the change in the mean with the opportunity of selection, you can also bound the variance for the opportunity of selection. If you don't have normality, then you've got this more elaborate expression, but again, it still gives you a bound. That's the quadratic selection differential. You take the observed mean, change in variance, and you adjust by adding the square of the directional selection differential. That then gives you a covariance term, and if that's a positive term, it means that fitness increases as you move away from some value. If it's a negative, move away from the mean. If it's a negative term, it means fitness decreases as you move away. And if you're worried about the mean, you can think about that as moving away from one point. So you take the optimal value. As you move away from that, fitness either goes up, disruptive selection, positive value, or goes down, stabilized selection, negative value. Just like we had a quadratic selection differential, we can have a quadratic selection gradient. And that's simply defined by this expression here. And actually, if you look at that, that's also a regression, a covariance, and this here is the variance of that term. So this gradient term here, like beta, that's the coefficient we see in fitness regressions with a quadratic term, and as we'll see a little bit later on, it's also a measure of the geometry. So I'm going over these somewhat quickly. As we talk about multiple traits, we'll come back and talk about S, beta, C, and gamma, and sort of put them all together. But again, the critical part is a selection differential is an observed within generation change. A selection gradient is something that in multiple terms corrects for correlations. We talk about directional gradients, directional differentials. We worry about change in means. We talk about quadratic gradients and quadratic differentials. We talk about changes in variances. Changes in variances are curvature. Changes in means are, is there linearity in that fitness surface? So these betas take on several different potential roles. We've seen that beta is the slope of the regression of fitness on phenotype. We've seen that beta is the derivative of the fitness landscape with respect to mean. If phenotypes are normal, we also have this other relationship, which is this is the average, this is the slope of the individual fitness surface at point Z. This is then the average slope. So if you have normality, beta is the average slope of the individual fitness surface. Beta is the gradient with respect to the mean population fitness surface, and beta is also the slope in a fitness offspring regression. Again, a lot of moving parts. It's going to be a little fuzzy. We're going to come back and talk about these again and again and again. So, if you're a little bit confused now, we're going to come back and sort of clarify these as we go through these more. But we have this notion. The key part here is directional and quadratic, means, variances, differentials and gradients. Differentials observe changes, may be corrected for something. Gradients, the actual changes when you account for correlations. For single traits, there's only a scale difference. For multiple traits, there's dramatic differences between these. Likewise, you can show that gamma is, this is the second derivative is the curvature. So gamma, if phenotypes are normal, is simply the average curvature of the fitness surface. So these beta and gamma have interpretations in terms of curvatures of individual fitness surface, regression coefficients on individual fitness surfaces, fitness landscapes. Since they're involved in fitness landscapes, as we'll see, they're involved in predicting how traits change. Does the mean increase or decrease? Does the variance increase or decrease? Yeah, we'll show that a little bit later on. Yeah, I've got a big table that puts all this stuff together. But rather than showing you the table, which would be just as confusing, we'll kind of work through these and go back and forth. So I realize there's lots of moving parts around. So what you should be taking in mind is directional, quadratic, differential gradient, right? Those partition. Then we've got different geometries. And don't worry about what does what, but think about geometry number one is what are the coefficients on that linear term or on that quadratic term for regression of relative fitness on phenotypes. Number two, that's the best fitting regression. It's not the actual fitness surface. Number two, what's the mean curvature, mean slope? Number three, what does it say about fitness landscapes? And beta and gamma are things that are involved in all three of these measures of geometry. And under certain conditions, namely normality, they're all equal. Under other conditions, as we see, they'll be a little bit different. But again, we'll come back and talk about that. So I can ask how traits change. This expression here is the Landy expression, and you see what it depends upon is the genetic variance, and the only measure of selection is beta. Once I have beta, phenotypes don't enter after that. We can also compute the change in variance. I haven't talked about this. I'll just give you an outline. Chapter 16 covers this. The change in variance is generated by disequilibrium, and you can write this expression here. It's called the Bulmer equation, and you can show that expression is simply equal to this. So the within generation change in variance, you can write as the quadratic selection gradient minus directional selection gradient squared. And what happens that's the within generation change. The between generation change is you keep this part and you lose half of the previous disequilibrium value. The key part here is that's the equation for change in the mean It depends upon the additive variance and the directional selection gradient. This is the expression for change in the variance. It depends upon the additive variance and now depends upon the quadratic and directional selection gradients. So gradients are the quantities that appear in equations of evolutionary change. So we can talk about measuring fitness surface. We can also talk about projecting how the mean and the variance are going to change over time, and the gradients, beta and gamma, are involved in both of those. So let's talk about one more thing, and we'll take a break and see how people are doing, because again, a lot of stuff, we're going to be talking about these concepts again and refreshing our minds about them. Here is the best fitting linear regression. That slope beta, that best fitting linear regression, remember the true fitness doesn't have to have this form. This simply says if I've got some relationship of individual fitness with respect to mean phenotype, what's the best fitting linear predictor for that? That best fitting linear predictor, the slope on that is given by the covariance of these divided by the variance of z. That's exactly the definition of beta. So beta is the slope on the best fitting linear regression. This basically says how much the variation fitness is accounted for. We can now consider the best fitting quadratic regression. So here it is. And I've made life easy by setting the trait mean here equal to 0 just to make life easy. So we now have two slopes. These slopes tell us about the local geometry. So for example, beta 1 is the average slope. The second derivative, 2 beta 2, is the average curvature. So in this expression, if z is normally distributed, that's given by beta, that's given by gamma over 2. If z is not normally distributed, as we'll talk about, that's different, that's given by gamma over 2. So the key part is when we have these regressions that approximate the true fitness function, the best linear approximation, that regression slope is given by beta. In the best quadratic regression, the quadratic term is a function of gamma, gamma over 2. So these gradients for the mean and the variance describe how the mean and the variance change over time, because they're functions of the fitness landscape. They also define they also are involved in coefficients on the best approximations with linear and quadratic regressions. Let me just make this point really quickly. So what happens, this is when you actually work out what these expressions should be, you get these results here, and this is assuming nothing about the distribution of z. When the distribution of z is normal, the skew terms here are 0, and these terms here, as we saw before, that's just 2 sigma squared z. In that case, beta 1 collapses to b1 collapses to beta, and beta 2 collapses to gamma over 2. If you don't have normality, this term here does not necessarily collapse to beta, and so if you want the best fitting linear term on a quadratic regression is not the same as the best fitting linear term on a linear regression if the data aren't normal. That has to do with skews. So a lot of technical details. Let me take a break here and tell you the good news is we're out of the technical details for a while. We're now going to look at actual data and see what do people say about the values of these. Let me stop back and give you kind of a big overview, the important stuff, because I throw a lot of stuff at you. We have changes in means and variances, directional and quadratic. We have differentials, directional selection differential, quadratic selection differential. Those are based upon the observed change in the mean, and the quadratic selection differential, the observed change in variance corrected by adding s squared. We then have gradients. Gradients are the things we want to work with. They have these nice features. Those features will be even nicer when we talk about multivariate space. So gradients correspond to measures of average slope and curvature. They correspond to expressions which are used in predictive equations. How's the mean going to change? How's the variance going to change? They also correspond to terms in the best linear regression of relative fitness on phenotype, and the best quadratic regression of relative fitness on phenotype. We're going to come back and talk about those points in a little more detail, about the geometry, so don't get too hung up on those now. We've got measurements of change in means and variances, observed measurements are differentials, measurements which adjust for phenotypic covariances are called gradients. So questions so far? A lot of detail, don't get too hung up on it. We're going to come back and talk about that, put it on the big table. More interesting question, of course, is, okay, we can do this machinery, what does it tell us? So how strong is selection in natural populations? So I mentioned Bumpus and Weldon were the first to publish attempts to detect selection on quantitative traits in nature. John Endler was the one that made the first serious attempt to put some of the early studies together, and then Kingsolver, along with people like Hopi Hoekstra and others, published a series of papers starting in the early 2000s where they basically summarized all these estimates of beta and gamma from natural populations. So following the 83 paper of Landy and Arnold, a massive cottage industry exploded. I think the Google citations are in the 2000s for this particular paper. Very influential paper. And the literature blossomed with all these estimates. And the field where you take estimates, a large number of estimates, to try to make general statements is a field called meta-analysis, basically the analysis of analyses. And there's several levels of sophistication you can apply. We'll talk about the simple one, which is called the narrative meta-analysis, where you just kind of present and show general features. More general things involve using mixed models, and Appendix 4 talks about those in detail. So here are some of the people involved. So John Endler is one of the people that put some of these early together. Joel Kingsolver, along with Hopi Hoekstra at that point. Hopi's gone on to do a lot of really interesting work on pigmentation genes in mouse. She's the head curator of mammalogy at Harvard. So these were authors and a bunch of others on that first influential paper, and then John was one that, in his early book from Princeton, talked about measurements of selection. And here's the sort of data you get. That star means I take character values which are variance standardized. We'll talk about mean standardization impacts a little bit later on. So what does this mean? What does a beta star of 0.5 mean? It means a 1 standard deviation in the trait changes relative fitness by about 50%. So it's got a nice, pretty clean interpretation. Here are the actual count data, and the book has a cleaner figure. This roughly follows an exponential distribution with the median value right there of being about 16%. This means that a 1 standard deviation change in the trait changes 15 by 60%. Selection is generally weak, with only 10% or so having gradients of about 0.5. So here, if you have more than 1 standard deviation, your fitness gets cut in half. And furthermore, most of these large estimates of beta occur in small populations. And the reason that's interesting, small populations have large sample variances, so these inflated values, these values may actually be inflated simply because of sampling error. For population sizes bigger than 1,000, most estimates of beta are below 1/2. So these larger estimates up here may be overestimation simply due to larger sampling noise in smaller populations. The more interesting one was the quadratic. So is that strong or weak? We'll talk about that. It's hard to say. I mean, depending upon your perspective, it goes very weak or very strong. The more interesting one is gamma. Now, in beta, the sign in beta doesn't really matter, right? The sign means you're either increasing or decreasing the mean, but the sign in gamma does. A negative gamma means you've got stabilizing selection. A positive gamma means you have disruptive selection. What do you see? It's symmetric about 0. We would expect, being evolutionary biologists, that stabilizing selection is pretty common, right? We talked about it before. We talked about testing for it in the fossil record. would generally expect that if you take a generic trait, that there's some optimal value. So if I select it far enough one way or the other, fitness starts to go down. So you expect that a lot of traits we see here are, relatively speaking, under some quadratic negative curvature. So they're under some type of stabilizing selection. Yet we see no signal of it. We don't see a strong shift to one side or the other. There are possibilities for why that's the case. The book talks about it a bit more than I will. One thing we'll talk about more tomorrow is Mark Loza and Rob Brooks pointed out there are issues in terms of how these terms are estimated that may result in a lot of them being underestimates. So let's look at then the importance of experimental manipulation. And here's a great example. Remember I talked about the invisible fraction? We showed that great example very early on of genotypes with large and small flowers, and that if you looked at those that survive, genotypes with large flowers have much more seed. But if you look at survival, genotypes that will have large flowers at some point have a much lower survival rate. That's an invisible fraction. A wonderful experiment is given by Barry Sonnero. What he basically showed was that clutch size in these side blotch lizards, there's the blotch on the side, has an impact early on. So he used hormonal control. He also went in and surgically removed some of the ovaries. So he'd have fewer ovaries, sewed them back up, and looked at the survival of those. And what he showed was there's a viability cost to increased clutch size that occurs before clutch size is expressed. So in those individuals that had ovaries surgically removed, they actually had a better survival rate than those individuals that didn't have ovaries removed. So it shows you the level that you have to go to ask some of these questions, but it also shows you the importance of this invisible fraction. And those were two examples that if you simply looked at the existing data without manipulation, you'd get very misleading conclusions. You can do two types of manipulative experiments. You can change the environment and ask what happens to fitness. You can change the phenotype and ask what happens to fitness. And these are complementary. So for example, Weldon, in his crab experiments, basically showed that he thought that the carapace shape, that is the shape of the shell, might have changed because the breakwater had been completed, resulting in increased silting. So to test this, he exposed crabs to silt conditions similar and found survivors in this laboratory setting had smaller frontal length, which is what he actually observed in Plymouth, and he thought that smaller frontal length was more important in terms of filtering out silt before it got into some of the feeding structures. So one of the earliest examples of using environmental manipulation to try to get a handle on what the causative site of selection is. Another example of environmental manipulation is the imported willow leaf beetle, and Mike Wade's done a lot of work on that. That name looks familiar. I think she's the department chair here. So Mike Wade and Sue Kalis looked at the imported willow leaf beetle, which is a gregarious species. And what they basically found is that there's a positive relationship between group size and fitness. So populations that have larger group sizes tended to also have larger fitness. However, when you removed predators, that relationship went away. So the environments are predators, no predators, and what you saw is a fitness relationship changed as you changed the environment. So those are both examples, the crab example and this of changing the environment. The classic example of manipulation is my favorite one, long-tailed widow birds, window birds in Kenya. So males here, please don't draw any conclusions in this. The thought was that males with longer tails are preferred by females. So what they did to check this is they experimentally manipulated this where they increased or decreased the tail size. And what they showed was males with artificially induced longer tails did indeed have greater mating success than males with normal or shortened tails. But even more importantly, they showed that males with shortened tails had no impact on the abilities of males to control territories, indicating it wasn't male-male competition, but rather female choice that was acting on the tail length. Really cool experiment. Another cool experiment is the ruby-spotted damselfly. You can see why it's so named. And what these authors did in a little pond in California was basically to ask, does this spot have any impact on mate choice or on fitness? So what they basically did was they measured reproductive lifespan, how long they survived in the mating era, mating rate, and lifetime mating success, combination of both. All three of these showed significant gradients on red spot, but not on body spots. So if you had a larger red spot, you basically had a larger reproductive lifespan, more lifetime mating success, and a higher mating rate, but body size didn't seem to play a factor. So to adjust for how important that was, they basically took individuals like this and they used a red pen and expanded the size of the spot. They also, very cleverly, took a clear pen and made the same area so they could test whether or not the pen by itself, independent of the color, changed things. And what they showed was that males with enlarged red spots had mortality rates that were about a quarter higher, 23% higher than control, while the sham and control groups had no difference. So simply adding stuff on there that was clear had no effect, but adding red did. So contrary to what the fitness suggests, direct manipulation shows that increasing spot size decreases survival. So there's other features that are giving you the observed strong gradient of survival on spot size we saw up here. It doesn't seem to follow when you do the manipulative experiments. So whenever you have a fitness estimation, you can think about that as an initial hypothesis to test. There are a number of ways it can fail, and these direct tests are actually fairly useful. A final thing are great tits here in England. There they are, cute little bird. And what they basically ask is, if you look at the selection differential on fledgling weight, and then look at that, and someone asked here over different environments. So they looked at mean temperature, and they also looked at beach mass scores. So mass score basically is how many acorns or how many seeds do you put out. And what they showed was as temperature and mass score changed, the differential on body weight changed. So this is an example, what's often called an ecophysiological surface, showing how as you change the environment, how you change the selection coefficient on that. And the last example is this great study on anolis lizards. And what they did was they had small islands in the Caribbean, really small. You can basically go in there, and on those small islands, you can basically remove predators and ask, is there an impact? So if you don't remove the predators, you remove the birds, remove birds and snakes. Stamina seemed to have no change. Hind wing length had no change. However, if you simply change the density and looked at high and low density, you see significant differences. So these features here, hind wing length and stamina, seem to be more caused by competition, differences in densities, as opposed to predation. So these last few examples just show you how you can use manipulation to ask kind of interesting questions when you think you found a fitness trait association. So that's the overview. I realized there was a chunk in the middle there that was fairly heavy when I was throwing all these concepts at you, like slope, average slope, regression coefficients, fitness landscapes. We're going to come back multiple times and tie all together a bit neatly. But the key feature here is you've got differentials and gradients. Differentials are observed changes. Gradients are the actual targets when you do appropriate corrections. Gradients are the things that appear in what matters. evolutionary equations to predict changes in means and variances, regression coefficients, average slopes, average measure of landscapes. We then talked about what the data say about average values of selection gradients. We'll come back and say a bit more about that tomorrow. And then we talked about a couple of examples of experimental manipulation.